Again, our lab review for lab test 3 will be on YouTube. After we review today, I'll make sure and post that on YouTube like I have before. Some people really like that and use that tool. Others of you don't even care. So here we go. I have exercise 10, human genetics. Remember, these questions would be referencing humans. Where are autosomal characteristics carried? Yeah, what are the autosomes for humans? The chromosomes that aren't sex. So let's just take a quick glance in our lab manual, page 123 at the top. Page 123 at the top is a human karyotype, and there are all of our chromosomes. So which ones are autosomes? Is number 1 an autosome? Yes, because it's not a sex chromosome. Is number 2? Yes. So which ones are autosomes? 1 through 22. 1 through 22 are autosomes, because those are the ones that are not sex chromosomes. Autosomes are not sex chromosomes. Okay, in humans, where are the sex-linked traits carried? 23rd pair. Very good. 23rd pair. Everybody okay on that? Okay, number 2. What chromosomal abnormality is associated with Down syndrome? Chromosomally, what's the problem? There's 3 what? 3 21st chromosomes. 21st. So, if you want a reference on Down syndrome, page 122 has a reference on that. Down syndrome is also called trisomy 21. It actually tells you what the problem is in the name, trisomy 21, Down syndrome. Make sure you can recognize a karyotype of an individual with Down syndrome. That'd be really easy to spot. You just look at the 21st pair, and if there's 3 chromosomes there, then the individual has Down syndrome. Okay. How can you tell the sex of an individual in a normal karyotype? Let's just practice. Go back to page 123 again and look at figure 10.3. Buddy, look at their 23rd pair over there on the bottom right-hand corner. What do you see? I see male also. What makes it male? X and a Y. And specifically, it's the Y. Denise is right. If you specifically see a Y, you've got a male. But generally, most males are X and Y. What about if you had 2 X's there? That would be female. So it's something you can do? Yes. Okay, what chromosomal abnormalities are associated with Turner syndrome? Well, on page 125, I found page 125. That was very useful. On page 125, we have a table here, table 10.1, where we talked about the chromosomal abnormality, and we used symbols to show what the problem was. I'll get us started with Turner's. XO. What does the O mean? There's only one X. The O really means 0, but they don't have another sex chromosome. So they're short a sex chromosome. So by default, they're female. Because you have to inherit a Y chromosome to be male. So this would be a female. You may remember that she's kind of got a webbed neck. She may have kind of a broad chest, little breast development, no mincing. She doesn't have a menstrual cycle. She's sterile. Okay, that would be Turner's. Okay, help me out with poly X. How could I show poly X? Okay, I heard 3 X's. Is everybody okay with that one?
Does that mean they're also female?
They are also female. Yes, ma'am. Okay, Klinefelters. XXY. And what would they be? Y. Because of the Y. That's funny. And then Jacobs. XYY. And what would they be? Male also. Okay, now really important is being able to look at a karyotype and tell me what the disorder is. We practiced this on page 125. There are 4 karyotypes right there in the middle of page 125. Make sure you can look at a karyotype and know what the disorder is. But okay, so we're just pretending. If I gave you karyotype A, everybody glance at karyotype A, and then your choices were these that are up on the screen, then you would have to pick. And what would you pick?
Poly X.
And why would you pick poly X?
Because there's 3 X's.
Because there's 3 X's. Now guys, let me be clear. On the test, I don't have all this pink stuff written on the test. The test's going to look like that, where it just says Turner's, and that's it. Or it just says poly X, and that's it. Does everybody understand? I know! So, I didn't want to mislead you, and you think that I'm going to tell you Turner's, parentheses, XO. I'm not doing that. You have to know that Turner's is XO. Okay? I know. Life's tough. Okay, number 5. If a person is homozygous dominant, you know, like they're big A, or they're heterozygous, like they're big A, little a, what characteristic will be shown in their phenotype? Dominant or recessive? Dominant. I agree. Dominant. They're just going to look dominant. You know, you really can't tell by looking at them if they are big A or big A, little a. All you know is that they're dominant. Okay, second part. If an individual is homozygous recessive, like little a, what trait will show in their phenotype? Recessive, definitely. Okay, that was question 5. Question 6, you would expect the dominant trait to be the most common trait in a population. Is this always true? No. Because you guys know, sometimes the dominant trait is not the favored trait, you know, the one that gives the best advantage in an environment. Let me just give you a quick example, because really a reason might be natural selection. Okay, so here's what you've noticed. I've noticed it too. I've noticed that people in their native lands, if they live near where they're native now, if they live near the equator, they have darker skin. Have you noticed that? Mm-hmm. Darker hair, darker skin, darker eyes. And then if they live away from the equator, they tend to be lighter skin, lighter hair, lighter eyes. Have you guys noticed that? Okay, so they were trying to figure out why. Thanks for coming. So they were trying to figure out why. So it's not hard to figure out that if you have dark skin and you live near the equator, you're being protected from ultraviolet radiation. Okay? But why is light skin favored away from the equator? It took them quite some time to figure out. It has to do with the production of vitamin D. If you live away from the equator, do you get as much sunshine as you do if you live at the equator? No. Okay, and if you have dark skin, I don't know if you know this, if you have dark skin, not as much sunshine penetrates your skin because of that darkness. That's what's protecting you from the UV radiation in the sun. Okay, but if you have light skin, more of the sun penetrates your skin. And so if you live far away from the equator, you need to be out in the sun because the sun is how you make vitamin D. You have to have sunshine to make vitamin D. Sunshine is good for us in small doses, not huge doses, but small doses. So it's actually an advantage to have fairer skin away from the equator. Yes?
Just in case we forget all of that, what you just said, could we kind of refer back to the lab we did with the bird thing?
Exactly.
Like the same thing?
It'd be about the same thing. It's natural selection. That's very good. So she's saying that when the bark of a tree is light, then the lighter peppered moth, which would be recessive, would be favored. Very good. So I'm just going to write here natural selection might be a reason why. That might be a reason why. There could be lots of reasons why.
What page was that lab?
It wasn't a page, it was a handout. That's right. Number 7, if I give you information about which trait is dominant and which trait is recessive, be able to work genetics problems. So, for example, guys, page 127. On page 127, at the bottom of the page, we did a problem about Nancy and her family. And then page 128, we did problems about Joe and Henry And let me see, Sally's on there. There's Nancy again. Gosh, there's all kinds of people on here. Henry. Boy, Henry showed up twice. So, sure. So, everybody take a look at, we'll just do something like, well, let me see. Can we work one? Oh, here's one for us to work. Yay. So, for example, can everybody look up here and tell how I was letting you know what's dominant? Take a look. 2 people with widow's peak, and look what's in parentheses. What is that? A big W. Okay, what does big mean? Dominant. Okay, so 2 people with widow's peaks have 3 children. 2 of their children have widow's peaks. 1 of their children has a straight hairline. Now, what would straight hairline be? Dominant or recessive? Recessive. What are the genotypes for each? So, let's see what you think. Okay. What, do you want to take a picture of that? Okay. All right, so I made a key at the top that big W is widow's peak, little w is a straight hairline. So go ahead and tell me, what do you think the parents are? Yes, they're big W, little w. Parents are big W, little w. I'm going to go back because it said the 2 people had widow's peak. So I know they have at least one big W, yes? But how do I know they're big W, little w? Yeah, because they have a kid that has a straight hairline. There's only one way that happened, and they had to be heterozygous. Okay, now, remember that they had 2 kids with widow's peaks and 1 kid with a straight. So let's talk about the 2 kids with widow's peaks. What would be their genotypes? They could be big W, big W. What else could they be?
Big W.
Yeah, we don't know. They could be either one of those. Not for sure. Because you can see in this cross, they could be big W, but they could also be big W, little w. We're not sure on those 2. But what about that other kid with the straight hairline? What for sure is their genotype? Little, little. The kid with the straight hairline for sure is little, little. Little w, little w. So that would be an example of what you would expect. So if you go back and read these questions and you can answer the questions that we did as a class, then you'll be fine. Just remember, I'm going to tell you what's dominant, what's recessive. You needn't memorize any of that. There's no reason to memorize that unattached earlobes is dominant. Okay, that's just silly. I would tell you, okay? All right. Number 8, true or false? Sex chromosomes carry only genes that relate to the sexual characteristic or sexual function of an individual.
True.
I heard both.
True.
It's false. So, I'll give you an example. There's a gene on the X chromosome. Does everybody agree the X chromosome is a sex chromosome? Yes? Okay. There's a gene on the X chromosome that determines whether I can see in color or not. Yeah. Does that have anything to do with my sexuality? No. So color blindness versus color vision, that would be a great example. That would be a great example. So thinking about X-linked traits. Remember that X-linked traits, this is question number 9, X-linked traits are traits on the X chromosome, like color blindness, or like hemophilia. That meant where your blood wouldn't clot, hemophilia. Okay. It says number 9, how are X-linked traits inherited differently, males versus females?
Females carry males.
Excellent. Okay, so females, that's weird. Females carry. I want to put that they're carriers. And then males show the disorder. So I want to remind you why that is the case. Remember that females are 2 X's, but a male is only 1. So a female gets 2 chances to inherit the dominant allele, the non-defective allele, But males only get one chance to inherit the dominant allele. They could also inherit the defective allele and have the disorder. We get two chances. So, for example, let's say that each of these individuals I have up on the screen inherited the recessive allele for color blindness. So this would be X little c, this would be X little c. Okay, but because this female gets another chance to inherit another allele, she could be big C, little c. Now, would she be colorblind? No. But I want you to notice she inherited the same allele that he inherited, but then she got another chance to inherit a second allele. He doesn't get that chance. Okay, where do little boys, this is a little boy, where do little boys get their X chromosomes? From their moms. It's very good. So that these recessive alleles come to little boys from their moms. So that's why Deanna said moms are carriers, and then they give it to their sons. Okay, number 10, pedigrees. You know, pedigrees were those little pictures of families. Do you guys remember some pedigrees had a circle and squares? Yeah. What does circles mean? Females. Circles are females. And what did the square mean? I'm sorry? Males. And what if you put a line connecting the circle and the square? No. They're mating. That's good. They are not siblings. They are mating. That's very good. We do not want those siblings mating. Nope. Okay, so everybody's okay on the symbols.
Wait,
All right.
Sorry.
Go ahead.
Isn't it where there's like things like circles and squares and opposite not siblings?
Yeah, so we'll do one in a minute just to clarify, Jordan. Okay, then guys, I want you to also remember that some of the time we color in an individual. See, I'm just coloring in that male. What does that mean if I color it in?
He's infected.
He's affected. He has the disorder that you're studying, whatever that disorder might be. That's the basic ideas, is that a circle and square linked are mating partners. Then you're going to drop down a line because, you know, if they're mating, we only care if they're mating if they have some offspring. Let's see here. Let's make them have 3 offspring. And then you color in the ones that are affected. Is that okay? Can you tell by the little bitty one that I've drawn up here whether this is a dominant trait or a recessive trait? Kaylin, you're right. She said recessive. Everybody look at why. The mom doesn't have it, the dad does, but what about the kids? They don't have it. So this trait, this green colored in trait must be recessive. Okay, does a pedigree show genotypes or phenotypes? Remember, genotypes are the actual gene content. Phenotypes are what they look like. What does a pedigree show? It shows you big A little a's and little a little a's? It shows that to you? You have to figure that out. What does it show you? Phenotypes, whether you're normal or you have the disorder. It just shows you phenotypes. They show phenotypes. Okay, lastly, you have to predict how a trait is inherited, just like we just did. We said that this was a dominant disorder. Now, to make sure that you know your choices, look on page 131. Page 131. There's a table at the top of page 131, table 10.3. Those are going to be the 3 inheritance patterns that will be your choices on the lab test. Now, everybody turn to that page because we're going to practice with this pedigree. I made a pedigree up here. So, Jordan, you're right. Here's where I've got the 3 kids and then who they mate with and then their kids, okay? And then also notice that I use Roman numerals to show the generations, but Arabic or regular numbers to show the individuals of the family. Is everybody okay with that? Okay, so here's your 3 choices for what kind of inheritance pattern I've got up on the screen. You could pick autosomal dominant, autosomal recessive, or X-linked recessive. Now, before you pick, make sure you look at the clues. Everybody look at the clues. What do you want to pick? I heard a nomination for autosomal recessive. How come it's not X-linked recessive?
Because the children have
Okay, what kind of children have it?
Boy.
Just boys?
No. Both of
Boys and girls. What if you'd looked up here and it's only in boys? Then it might be X-linked recessive. Remember, X-linked recessive shows up mostly in boys, yeah. But I'm seeing it in boys and girls. Okay, now, why did you pick, I agree with you, first of all, but why did you pick recessive in the first place? Neither of the parents have it. That's good. And only one out of the 3 kids have it. That's excellent. So we picked recessive, then we picked autosomal recessive because we're seeing it we're seeing it show up in both sexes. Everybody okay on that one? All right, let's just use the letter A for the trait. We're going to do a few of these, try to figure out a few of these members of the family. I'd like to do generation 1, individual 1. Everybody look up there, generation 1, individual 1. What sex is that person? Female. Everybody found her? Here she is right here. Generation 1, individual 1. She's right here. Okay, what is her genotype using the letter A? She is big A, little a. She's big A because she doesn't show the trait. The trait's recessive. She doesn't have it, so she must be dominant. Why is she big A, little a? She's a carrier. How do you know she's a carrier? Because of which kid? Yeah. What is that, a boy or a girl? That's a boy. Yeah. So because of her son, you can tell she's big A, little a. Okay, now let's talk about generation 1, individual 2. That's her husband, right? Or whatever. And so it's her mating partner. So what is his genotype? Sure enough. Big A, little a. It's the only way this works. Agreed? Okay, let's try another one. I like their firstborn daughter. Generation 2, individual 2. Everybody do generation 2, individual 2. I'm ready. Okay, big A or little a?
Little A. Oh, big A, I mean.
2 big A's?
No, just
Big A, little a. How do you know for sure?
because it gets passed on to the...
Because of her daughter, yes? Everybody agree she's big A, little a? Okay, let's try another one. Are you ready? We're going to do generation 3, individual 5. Generation 3, individual 5. Go ahead.
Big A.
This one right here?
little a, little a.
Make up your mind. I heard big A, or little a, little a. What do you want?
Little A.
I'm going little a, because she has the disorder, right? And you said the disorder was recessive. Okay, I got one more. Generation 2, individual 5. Generation 2, individual 5.
Big A,
Big A?
Yeah. Or big A, little A. It has to be the carrier.
Yep, she has to be a carrier. She has to be. What's her husband? Little a, little a. So now you could for sure tell me what generation 3 individual 4 is. Generation 3 individual 4, this person right here. Generation 3 individual 4, what is it?
Big A, little A.
Has to be. No other way. But I feel okay about pedigrees? It's pretty good stuff.
How long is this test going to be?
It's 50 questions, just like the previous questions. I'm sorry, previous tests. Same format. Okay, Susan has blood type A. I love blood type problems. Susan has blood type A, and Jack has AB. What are the possible blood types of their children? Well, guys, it's been some time since we talked about blood types, so I made this little chart. I found it in your lecture notes. Made this little chart reminding you that blood types are actually phenotypes, and that if you have blood type A, which I'm assuming many of you in this class have blood type A, it's pretty common, that there's 2 possible genotypes for blood type A. Big A, or big A, O? Can everybody take a look at my table up here? I want to ask you some questions, just what's on the screen right now. Would you say the A allele for blood type is dominant or recessive? Okay, here's the question. Would you say A for blood type is dominant or recessive?
Dominant.
Definitely dominant. Would you say B is dominant or recessive?
Dominant.
Definitely dominant. Would you say O is dominant or recessive? Recessive. Right, check it out up there. A is dominant, B is dominant, O is recessive. All right, now let's check out our problem again. With Susan has blood type A, and then Jack is AB. We have to figure out what the possible kids' blood types are. Now on Susan, everybody, what do you want to use? Do you want to use big A, or big AO for Susan? You're going to use big AO. You're going to assume that she's heterozygous just to make sure you get all the possibilities. Is everybody okay with that? All right, great. So we're going to work it. I'm going to go ahead and put that O in here because you told me to. And then let's see what Susan and Jack could do here. Now, this is going to be one of those choose all that apply. This has multiple answers. So, would you choose that Susan and Jack could have a baby blood type A? Yes. So I'd color that one in. Can Susan and Jack have a baby blood type B? Yes. Can Susan and Jack have a baby blood type AB? Yes. Can Susan and Jack have a baby blood type O? No way. There's no way. So don't color that one in. No way. Everybody okay on Susan and Jack? Anybody have a question about exercise 10, 1 through 11? Anybody? Okay, exercise 11 is DNA biology and technology is the name of the exercise, and everyone, that starts on page 135. Now, class, you can find these definitions in your lecture notes, which that would be great, but I'd rather you apply them, so I drew this. You may recognize this. I drew it on the board downstairs before. It's what we call the central dogma, but what's most important is it is a brief description of how information flows through the cell. Each arrow up here stands for one of the three words we just had. Those three words are number one. If you'll just glance back, they're number one under exercise 11, DNA, biology, and technology. The three words are replication, transcription, and translation. We just have to figure out what word goes with what arrow. Okay, so the first one shows DNA copying itself and making DNA. So what would that be? Replication. Where DNA uses its own DNA template and makes more DNA. That one okay? Okay, then DNA pulls apart and RNA is made from it. So this is the second arrow where the information in DNA is used to make RNA. What is that one called? Transcription. Good job. And then the information in the RNA is used to make an amino acid sequence or protein. What is that called? Translation. Good job. But it'll come that one. And if you want word definitions, feel free to look those up. They are in your lecture notes or in your textbook. Okay, I have number 2. What are the 3 parts of a DNA nucleotide? Okay, well, I'm going to draw them, and then we're going to write what they mean. I hope this looks semi-familiar to you. Okay, so the P with the circle around it, what does that stand for? Phosphate. Nice work. Phosphate. Okay, the box with the S in it. Sugar will not be a choice on the test. Deoxyribose. Good job. Okay, B with the box. Bases. Okay, now these are the bases. Now let us chat briefly about the 4 possible choices of base that you have in DNA. What could this be? Do it again. Adenine, A, adenine. Cytosine. The G, I don't know. Guanine. Thymine. Good. But remember, those are your 4 possible bases in DNA. Everybody's okay? Yes.
Do we just need to know the letters, or do we need to know what the letters represent?
I think I use the letters. Everybody okay on this one? Okay, the second part. So, to me, DNA looks like a ladder, a twisted ladder. It is twisted into a helix, but it looks like a ladder. And so, assuming DNA looks like a ladder, what parts make up the sides of the ladder? And here's your choices. Is it the phosphate, the sugar, the base? What parts make up the sides of the ladder? Phosphate, anything else? What else? Not sure? Okay, let me show you a picture of that ladder. You recognize this? What is this a model of? DNA. Okay, what are the white tubes? You see how I have white arrows on these things? What are the white tubes? Phosphate. So this is the P, the circle around it. What's the black pentagon? Deoxyribose. So this is the sugar. So it looks to me like the sides of this ladder are not just the phosphate. What is it? And deoxyribose. So I'm going to go back to the previous question. It's the phosphate and the deoxyribose make up the sides of the ladder. The sides of the ladder. Okay, let's look at that picture one more time. There's the sides of the ladder. Okay, does everybody see the steps, also known as rungs of the ladder? What's making up the steps of the ladder? The bases. So I'm going to go back there. It's the bases that make up the rungs. Now, glance one more time. It's not one base that makes up a step. It is 2. So let's talk about those bases. Someone told me earlier one of the bases was adenine. Who does adenine always base pair with? Thymine, T. That's good. Always. And who does cytosine always base pair with? Guanine. Very good. Guanine. Now, if you'd like some better artwork on that, page 136 is good. But the actual model that you and I built, that's what's going to be on your test. You may have already taken pictures of these. That day that we were working, I was encouraging you to take pictures of the models with your cell phones. If you look back at your photos, you may have something just like this. I see one arrow up here that we did not label. Buddy, check it out. It's that solid white rod that's holding the bases together. What is that? What kind of bond? No, not peptide. Hydrogen. Very good. This is a hydrogen bond. Everybody okay on that?
Picture, are you gonna put a picture like that when we have to label it or no?
No, it's the actual molecule. So it's the actual thing. Okay.
What page did you say is good?
Page 136. Okay, number 3 says, if I give you a sequence of DNA bases, be able to predict the base sequence on the other strand. So I thought, let's just practice. So I threw a little piece of DNA up here, and I want you to help me write its complementary strand. Questions, Samaria? Okay. You're stretching. Good job. Good job on that stretching. Okay, so help me out. Look up at the screen. This is 3', so what would be down here? 5. And then go ahead. A. Yes.
We have to like do that thing, like that coding thing.
Uh-huh. Like we're going to do it in a minute.
Oh, sorry.
Mm-hmm. Now we did practice this in class on page 138. If you want to look at what you did on page 138. I don't know. I could hear a few of you telling me what to do. I don't know if all of you could do this. I'll know after Thursday, but I don't know today. Okay, so you might want to go back and practice that. Page 138 is where we practice doing this same thing, which is called complementary base pairing. Okay, number 4, what are the differences in structure and composition between DNA and RNA? So look, guys, I want to make a table here. I'll put DNA on one side and RNA on the other, and I'm going to come up with 3 differences. 3 differences in structure or chemical composition. Who can get me started?
Double helix.
We have urine.
I mean, you know what I mean.
Uracil. Thanks for participating, Kaylin. I don't mean to make you feel bad about that. Yes, thymine and DNA, uracil and RNA. That's good. I need another one.
DNA has double helix.
Okay.
RNA single strand.
Excellent. Okay, I've got that DNA's double-stranded, RNA's single-stranded. I need one more thing. Good. Lindsay said that DNA has deoxyribose for its sugar, and RNA has ribose for its sugar. Everybody okay on that? You can do that table? Sweet. So now I have another picture of another model that we built when we were we were working that day. Does anybody know what is this molecule?
RNA.
It is RNA. What kind? It is mRNA. Who told me that? Good job, Lindsay. mRNA. This is messenger RNA. mRNA. Okay, now we got to label the parts that are have white arrows on them. Let's just start with this first one. It's on the white tube. What is that?
Phosphate.
Yep, still is phosphate. How about the red? That's a base. And how about the blue pentagon?
Rose.
That's ribose, the sugar. Is okay on this one? Yes? Because this is what's on your test right here. Okay, what's a codon? I'll get us started. It's 3 bases on mRNA. It's 3 bases, 3 right in a row, bases of mRNA. And then, like, what's it for? So the question is, what's its function? What do those 3 bases do? They code for something. What do they code for? Not a whole protein. That's good, Kaylin. They code for 1 amino acid. And here's all their codes right here. Have you seen this before? Me too. It's called the genetic code. Is it going to be on our test? Yes. Do we have to know how to use it? Yes. You're going to practice in just a second. So let's dig through our lab notebook or wherever you keep your stuff, if you keep your stuff, and see if you can find it. It's called the genetic code. Everybody dig theirs out. Dig, dig, dig. Genetic code. Is there anyone who cannot find it? Come on up. You can borrow one from me. If you cannot find one, come on up. Get your genetic code. Come get your genetic code. Okay, to finish up number 5, what organelle is associated with the process of translation? It is the ribosome. That's good. Ribosome. The organelle associated with translation, the little cellular part that helps make protein, is called the ribosome. You may remember it's made out of little r RNA. Okay, so number 6 wanted you to be able to take a piece of DNA and give me messenger RNA. So I thought, let's just practice. So I put a little piece of DNA up here. How do you know what I have up here is DNA? Because of the T's. And how do you know what I want you to do is give me RNA? Transcribe. The word transcribe tells you that I want you to make RNA. But okay with that? Okay, so get me started. Here's 3 prime, so what do I put down here? I'm sorry? 5. Okay, go ahead. No. A. U.
G.
Good.
C.
Go.
G. C. U. A. U.
Is Diamond the only one that can do this?
U.
Okay, what's the last one?
U. A. G. 3.
It seemed like Diamond was the only one that could do this. Erica, can you do this? Okay. Right, so guys, check it out. That's a codon, that's a codon, that's a codon, that's a codon. Okay, on what a codon is. And you can transcribe. We did practice this. Yes, we did. We practiced transcribing, page 141. If you want to look and see where you practiced transcribing, then you did on page 141. Okay, I took another picture. I remember making you do this on your tabletop, and I came by and checked it. Got all kinds of stuff up here. First of all, I've got a process being simulated here on this tabletop. What's the process that's shown here?
tRNA. Translation.
So a person might ask, how do you know you're doing translation? Well, I'm just going to take a look back at what you just said. What organelle is associated with translation? Well, look at it right there. Do you see it? What is that? That's a ribosome. If you see a ribosome, you're doing translation. Because you just told me that the ribosome is used for translation.
And the abbreviation translations are RNA, right?
The abbreviation for translation.
Well, like when it's like mRNA messenger.
Oh, this would be rRNA. You're right. Okay, now I'm going to redraw this arrow. Now I want to know, what is this whole big thing up here that's got like this white and blue? What is that?
mRNA.
That one's mRNA. Okay, so guys, mRNA is this whole thing right here. That's all mRNA. mRNA. Okay, then we've got these 3 things right here. Do you guys see my 1, 2, 3. See those 3 things? What are they? That's tRNA. That's good. tRNA. So I see that translation involves a ribosome, which is rRNA. It involves the information mRNA, and it involves the taxicab driver, which is tRNA. All the RNAs are involved in translation. Everybody okay with that? Okay, good. What did I draw here? I'm sorry, take a picture of here.
tRNA.
This is tRNA. This whole thing is tRNA. No, I don't want that there. Put it over here. Okay, there's some parts to tRNA. Let's do the top part. I'm talking about these 3 things right here. What are they? That's very good, Haley. Bases of the anticodon. That's the anticodon. Well done. And what about this black thing down at the bottom? Amino acid. Well done. And don't forget, this whole thing is tRNA. The whole thing. The whole thing is tRNA. It's the whole thing. You guys okay with that? Yes, ma'am.
Will we need to know the individual names of the bases?
No. So, number 7 has to do with what we just looked at. Okay? So, this thing right here. That picture right there. That's why I put that picture there. Number 7. What 2 components necessary to do translation are carried by the tRNA? The what and the what? The anticodon. What else? And the amino acid. So going back to number 7. What 2 components necessary for translation are carried by tRNA? The anticodon and the amino acid. But okay on that part. Then it wants to know, what is an anticodon? I'll get us started. 3 bases. Ready to agree with that part? Complementary to a codon. 3 bases complementary to a codon. So do you think you're going to have to give me anticodons? Sure you are. Like this. I gave you codons. You have to give me the anticodon. Let's practice together. My first codon is CGC. What's the anticodon? G. Huh? C. Then G. But okay on that anticodon. How'd you do that? Complementary. You guys know G goes with C, right? That's called complementary. Complementary. Okay, do the next one for me. Go ahead. A, U, and what? A. Because you know A goes with U. Is it okay with that? We practiced that, everybody, at the top of page 143. If you want to look back and do some more practicing, top of page 143. We practiced making anticodons. Okay, so earlier, Kaylin said, am I going to have to use my genetic code and do the amino acid sequence? And of course the answer is yes. So we have our genetic code out. I want you to practice with the 4 codons. These are codons. Practice with the 4 codons I have on the screen right now. When you finish, check your neighbor. Okay, what did you get for AUG? Methionine? MET? ARG? Arginine? Okay, the next one? ISO.
My bad.
You are bad. TYR. Tyrosine. I'll agree. And the next one? Stop. You are. So is that okay? Everybody can use the genetic code. Don't forget the genetic code will be given to you. And I would put this with number 8. Number 8. Okay, we need to dig in our lab stuff again, and I'm looking for a piece of paper, everybody, that says electrophoresis. That's what it looks like. It's a handout that I gave you during lab one day. You should have kept it. It says electrophoresis. Let's look and see if we have that. Electrophoresis. Electrophoresis. Let's all dig for that. On the back of your electrophoresis sheet, you should have drawn a test tube and then labeled the 3 components inside the test tube. This test tube represents our procedure called DNA isolation. I think you probably labeled it DNA isolation. Is that correct? Yes? Okay, great. All right, what role did each play? The cell extract. What role was the cell extract playing in this procedure?
That's our DNA, our source of DNA. You're not right. It's not happening.
Wonder what I did wrong. There it goes. Okay, how about SDS? Dissolve the membranes. That's good. We have to dissolve the membranes to get the DNA out of the nucleus and thus out of the cell. And then the last thing we added to the test tube was the ethanol. What was the role the ethanol played? Separated the DNA. It's like a DNA magnet. Everybody okay on that one? Great. Now keep that piece of paper out and let's go to the front of it. First sentence. It says, what is gel electrophoresis used for? In the first sentence it says, gel electrophoresis separates molecules. It's used to separate molecules. That's what it's used for, to separate molecules. Okay on that one? Stay on the first sentence. Let's keep going. Upon what basis does gel electrophoresis separate molecules? Charge and size. Excellent job. Okay, last question on this one. What makes the molecules migrate? I got an E up there. Anybody help me? Electricity, good. You may remember we had to turn on the power supply and plug it in. Yeah. We have to have some electricity. Okay, on number 10. Super. Number 11. Okay, so it says if I give you a protein's isoelectric point, and if I give you the pH of the buffer, determine if the protein will be positive or negative. Well, guys, we practiced that on the front. Everybody check it out. Middle paragraph. Each protein has an isoelectric point. That is the pH at which the protein has no charge at all. In solutions with a pH higher than the isoelectric point, the protein becomes more and more negatively charged. In pH solutions lower than the isoelectric point, the protein becomes more and more positively charged. And then we practice that on that table. Yes? Yes? So go back and practice again. Go back and look at that table and see how you put the positives and the negatives and why you did that. That's what I'm talking about on the first part of number 11. Reread and re-practice that table. Second part of number 11. Determine the distance proteins will travel relative to other proteins. So let's just pretend that you were given this gel. Right, take a look at my gel up here. Pretend it was laying on a paper towel. Remember on the paper towel we put a negative sign and a positive sign? Now, just pretend that you had these 5 proteins, and I did them in 5 different colors so you could see them and tell they were different from each other. Okay, check out this question. Which of these proteins are positively charged? Choose all that apply. Who said 1 and 5? Who said that? Lindsay's right. It's 1 and 5. How come 1 and 5 are positively charged? Because they move to the negative. But okay on that question. You like that question? Okay. Deanna's correct. If they're positive, they move toward the negative. If they're negative, they move toward the positive. That's good. Good job. Okay, now, want to remind you, an isoelectric point is the pH at which the protein has no charge at all. So check out my second question. Did you see my pH of the buffer that this came from? Look up at the screen. What's the pH? 7. See the 7? pH equals 7? Okay, so check it out. Which of these 5 proteins has a pH, excuse me, it's wrong. Which of these 5 proteins has an isoelectric point close to 7?
4.
4 is correct. Who said that? Good job. Why'd you pick, Erica, why'd you pick number 4? It's in the middle. It didn't move very far. See, my black lines indicate where they started. And it didn't move very far. That means it didn't have very much charge. If you are close to the isoelectric point of a protein, it won't have very much charge, so the electricity will not move it very far. Kaylin?
So, are they asking us what it is before they move or after?
After.
Okay.
That's good. So this, I'm just putting a pink box around it. That's where they were at the beginning of time. And then about 30 minutes later, after I plugged in the electricity, that's where they are now. Okay? Good. So you guys remember you loaded them right into the middle? Okay. Anybody have a question about my pretend gel and my pretend questions? Okay, great. All right, we get to go to the back of our handout. Made it to the back. And on the back, it's called Evidences of Evolution. What is evolution? Here's my favorite definition of evolution. I'm sure it's your favorite too. We're looking for the genetic change in a population over time. Genetic change in a population over time is evolution. You can look that up, guys, in your lecture notes. You'll find the same thing. It'll be at the very beginning of our chapter on Darwin and evolution. But okay, on evolution. It should seem familiar to you. We've used that definition before. Okay, how does evolution explain the unity of life? Let me explain. So all semester I've been telling you all cells have DNA, and no matter who you are, A goes with T, C goes with G, and that DNA is double-stranded and twisted in a helix, or that all cells have RNA, and not just one kind of RNA, 3 kinds of RNA, mRNA, tRNA, rRNA. And all cells have ribosomes, and all cells have the same 20 amino acids, and all cells use ATP for energy. Do you remember all that stuff? Well, I call that the unity of life. When all cells have these things, I call that unity. So how does evolution explain why all cells have DNA? Why do all cells have DNA? Thank you. Common ancestry. Okay, we were kind of talking about this in lecture today, the diversity of life. I was telling you there's over 2 million different species of organisms, extant organisms. And so that's diversity. It means lots of different. So even though all life had one common ancestor, why are there so many species on Earth today? Say it again. Changes due to environment. That's good. So they're adapted to different environments. Very good. Very nice, Denise. So if the whole Earth was just one environment, we wouldn't have the diversity that we have on Earth today. But we have so many different environments. Thus, organisms have become adapted to these different environments. Well done. That was question number 1. Okay, question number 2. Everyone, you can find these in your lab manual, page 150, page 150. But you may remember these already. Paleozoic. Paleozoic. Does anybody know what the word Paleozoic literally means? Very good, Diamond. Ancient life. Ancient.
ANCIENT.
Is that right?
Yes.
I don't know why, it just fell out of my brain real fast. How about Mesozoic?
Middle life.
Middle life. Is Diamond the only one who knows these? Okay. Okay, how about Cenozoic?
Recent.
Recent. Oops. Page 150 if you want a reference on that. Okay, you're going to have to know the fossils. Where can you go to study the fossils? Blackboard. Under course content, there should be a PowerPoint with fossils. And you have to know fossils 1 through 12, and you have to know the name of the fossil and its era. Name and era. So you have to spend some time studying that. Okay, homologous, I found that word. Page 154. Top of page 154. Okay, I'm going to give you 2 choices for homologous. Is homologous same structure or is homologous same function?
Same structure.
Make up your mind which one. It's structure. Good. This is what your lab manual says. Parts of organisms are said to be homologous if they exhibit similar basic structure. So same structure. Or similar structure. We don't count homologous? Well, that's there. Analogous. What does that mean?
Similar function.
Similar function. Good job, Erica. Does homologous or does analogous represent a closer evolutionary relationship? Homologous, good. Everyone, we worked on this word homologous on page 155. You and the people on your table compared the forelimbs of many different vertebrates to the ancestral forelimb in the center, and we noticed similar structure. There were some variants, like the ulna and the radius were fused on the frog. They were not in our ancestor. But we basically saw similar structure in all those vertebrate forelimbs. That would show homology because it's similar structure. Definitely is not analogy because you guys know a frog is not using its forelimb the same way a bird does. They definitely don't have similar function, but they do have similar structure, so that would be homologous. Okay, next page in our lab manual, page 156, we compared the basic skeletal structure of a human to the basic skeletal structure of a chimp. And for this question, I just want you to think of what accounts for the differences from the neck down. So don't think about the skull right now, just from here downward. That's good, Denise. She said it's posture. The answer is posture. Here was the question. What accounts for the many differences between chimp and human skeletons? It's our posture, because we're standing up on 2 feet and they're on all 4. You guys also know that chimps are still able to go up into the trees, whereas we can't do that, or not well. And we're adapted for long-range walking where they're not. So our changes in our skeleton are due to our posture and just using 2 feet. Humans are walkers, living walkers. The walking alive. Okay, next question. So bad. Page 158. Page 158 are the skulls. I want 2 reasons why our skulls differ from chimpanzee skulls. Diet. They're mostly herbivores. We're omnivores. Brain size. Very good. We have huge brains.
That's why we have big heads.
We do have big heads, yeah. Just even me saying we have huge brains shows my big head. That's true. That is true. Okay, I have know the animal skulls in the lab. You guys have been checking out the skulls, right? And they also have a PowerPoint on the skulls, but feel free to take your own pics if you want to. All right, and then what do teeth tell us about an animal's diet? Let's say that I gave you a skull and all the back teeth were worn down so far you could see inside the tooth. That's an herbivore. That's good, an herbivore. What if I gave you a skull and you looked at the teeth and all the teeth were sharp? Carnivore. What if I gave you a skull we had some sharp and some worn down? Omnivore. Those are the 3 words you need to be able to use on those skulls back there. But okay, on the skulls. Okay. Okay, comparative embryology. Everyone, we did that on page 159. Page 159. How does comparative embryology help determine relationships between organisms? Okay, well, guys, if you'll just glance on page 159, I noticed that a fish embryo does not resemble a human embryo as much as a reptile embryo resembles a human embryo. Right, check it out. The fish looks more different than the human than the reptile. That's page 159. Do you agree with me?
Yes.
So what does that mean about our relationships? Who are we more closely related, the fish or the reptile? How do you know by the embryos?
Just similarity.
More similarities. So the more similarities you see, the more closely related they are. The fewer similarities you see, the less related they are. That just makes sense, doesn't it? Are you okay on that idea? Now, I really like this next part. Really, really like this one. Would you expect a human embryo to be more like a chick embryo or more like a pig embryo? Pig. I like your answer, pig. But we have to say why. Because they're bacon. What? Okay, chicks are birds, right? Go ahead. Denise is right. We are both mammals. Humans are mammals. Pigs are mammals. But what about chicks? They're not mammals. What are chicks? Birds, which are a type of reptile. That's very good. Okay, so because pigs are mammals and humans are mammals, we would expect the pig and the human embryo to be more similar than a chick and a human. Is everybody okay with that? I like these types of comparative questions. Comparative questions. Okay, number 8. I need some basic biochemical molecules all organisms have in common. If you don't know, page 160, top of the page, will tell you some. I know one right off the top of my head. Starts with the letter D. Go ahead. DNA. Does anybody know any more? ATP. Anything else? Many enzymes. I'll just give you some examples. ATP synthase, DNA polymerase, RNA polymerase. All cells have those. Okay, I don't think your book mentions this one. All the kinds of RNA. All cells have all 3 kinds of RNA. All cells have that. All cells use the same 20 amino acids. And you guys know this, all cells use the same genetic code. You know that piece of paper we had out a few minutes ago? So their codes are all the same. It's like they're all speaking the same language because they're using the same code. Isn't code language? Mm-hmm. So we're all speaking the same language, our cells are. That's cool. But okay, on number 8, okay, how could you explain that? Why do all cells speak the same code and have all the same chemicals? Good job. When in doubt, choose common ancestry. Some of you remembered that. Did you, Erica? Did you remember that?
No, Denise is always repeating after you that. She said, when in doubt, when in doubt.
That when in doubt. Denise actually listens. Isn't that amazing? Everybody okay on that one? Okay. Number 9. Let me show you some visuals to help you remember. One day, we put liquids from dropper bottles into these spot plates. First, we put rabbit serum. Then we sensitized the rabbit serum to human serum. And then we put serum from 5 different animals. And we had to figure out which animal was which based on their relationships to humans. Does anybody remember doing this? Yes? Okay. All right, here's what you're going to have to do. I'm going to set the same thing up for you. Now, I want to tell you that there's rabbit serum in there that was sensitized to humans. And you're going to have to tell me which of the animals on the 5 spots is most closely related to humans. How will you know? Lots of white stuff. If you see lots of white stuff, that's the most closely related. How do you know which one is the least related? No change. You don't see much white stuff at all. So lots of white stuff is closely related. Not much is not closely related. Everybody okay with that? Okay. Okay, last thing. You should have somewhere a piece of paper with all your fossils on it. Let me pick it up and show it to you. You guys still have that, the piece of paper with all your fossils on it? Okay, the back side of that has the cytochrome C. Cytochrome C. So I'm going to give you this, because you wouldn't have to memorize that. That would be silly. I'll just give you this thing in the middle, the cytochrome C clock, and then ask you questions. Specifically, I like questions like number 3. Number 3 is my favorite question out of all the questions that are on here. I like question number 3. According to the figure, have rattlesnakes and tuna fish been evolving separately for a longer time than humans and horses? You guys know why I like it. Because you had to look up 2 different numbers, compare the numbers, and come up to a conclusion. So it's the hardest question on the page. That's why I like it. Because that's what kind of teacher I am. Yes. So if you can answer the questions and use the cytochrome C clock well, then you're fine.
Can you answer the question?
Yes, the answer to the question is rattlesnakes and tuna fish. Okay, so we are getting close on time. I have about 5 minutes left, so I'm going to kind of dash through this last part. I am going to defer to your lecture notes on these definitions. Class, these definitions are on the lab test, so make sure you go back and look them up. Okay? And in your lab manual, if you'd like a page number for these definitions, they're on page 165 and page 166, if you need that out of your lab manual. Sorry. 165, page 165 and 166. But I have to defer to your lab manual. I do love question number 2. What is the purpose of doing Hardy-Weinberg? It is a baseline.
Baseline of evolution.
Okay.
None of evolution is occurring.
It's the baseline, so you can know if evolution is occurring. It's very good. Good memory on that. And you have to know this. Why do biologists do Hardy-Weinberg? Baseline for evolutionary comparisons. In the absence of outside evolutionary agents, how would you expect gene frequencies of a large population to change over time? Okay, I'm saying no evolution. What would you expect to happen to P's and Q's if there's no evolution? It'd stay the same. It's good, Denise. Because there's no evolution. And evolution by definition means the genetics are changing. So if there's no evolution, the genetics are not changing. Oh, I love number 3. Yes, yes, yes. I love number 3. Everybody take a look at number 3. This one's true or false.
Go back up.
True or false? The frequency of the recessive phenotype. That means how often you count individuals that are recessive, how often you count them, equals the frequency of homozygous recessive genotype. That means the percentage little a, little a. Why is that false? Okay, so who's got blue eyes in here? Anybody have blue eyes? Raise your hand if you have blue eyes. Okay, so I got 3 people in the class with blue eyes, so that would be 3 people divided by the total number of people in the class. Wouldn't that be percent homozygous recessive?
Yes.
Because you wouldn't have blue eyes unless you were recessive. Does everybody know blue eyes are recessive?
Yeah.
This one's true. This one's true. Okay, now question 4, similar but different. Look at question 4. What makes number 4 different? What's changed? It says dominant this time. The frequency of the dominant phenotype in a population equals the frequency of homozygous dominant genotype. So all the rest of you guys in the class either had green or brown eyes. Is that true? Okay. So that would be everybody else. So would that mean everybody else is percent little a, little, big A, big A? No. Why not? You could be big A, little a. So is this one true or false? This one's false. Okay, so if you're recessive, you are for sure little a, little a. But if you're dominant, you are not for sure big A, big A. But you see, that's what number 3 and number 4 are talking about. It's just very wordy, so it's hard to understand. That's really what they're talking about. Okay. Okay, I've told you this so many times, number 5, that I'm not playing around. You have to know the symbols of Hardy-Weinberg and what they stand for. Page 167, top of the page. We were talking about that downstairs, that you have to know. What do we call, in Hardy-Weinberg, the frequency of the dominant allele? P. You have to know that. I'm not making it up. So make sure you memorize what each symbol stands for. Okay, let's just pretend. This is number 6. Let's say that P is 0.5 and Q is 0.5. Everybody notice they add up to 1? Okay. Let's say I give you these genotypic frequencies, and then I say, what would you expect the P's and Q's to be the next generation if the population is obeying Hardy-Weinberg? Well, Hardy-Weinberg predicts no evolution. If no evolution is occurring, then everything stays the same. So if a population is obeying Hardy-Weinberg, it's an equilibrium. What does that mean? Staying the same. So what's the answer? 0.5 and 0.5. Well, hold on. What if you waited 5 generations? What would P and Q be? 0.5 and 0.5. They'd be the same. What if you waited 10 generations?
Same.
The same. So the answer is the same, the same, the same. If it's obeying Hardy-Weinberg, everything stays the same. Okay, this is the Hardy-Weinberg we did downstairs. You guys recognize it? So this one's in your lecture notes. Did that. So I'm going to jump over to the difference between the founder effect and the bottleneck. Okay, what's founder effect? Starting a new colony. Okay, so whatever genes those new colonists have, that's going to set the allele frequencies for the new population. Starting a new colony is how you could think of the founder effect. That's very good. Okay, what's bottleneck?
Survived by
Something
chance.
happens and you survive.
A catastrophic.
Catastrophic event, that's right. So you got a big population, something happens, and then you have a small population, the survivor setting the allele frequencies for the next generation.
Survived by chance, but with bottleneck.
And a bottleneck, so you got to have a big catastrophe where lots of people die. So that would be like, oh, I got it. Let's say we were all down in the lecture room and a tidal wave came in and completely covered Houston. Those people that were on the second and third floor, they would survive. The rest of us would be dead. And they were on the second and third floor just strictly by chance. Good.
Those are genetic drift, right?
Those are examples of genetic drift. Okay, here's our last question. I need some examples of natural selection pressure. I'll get you started. Predators. I'm thinking constantly in fear of being eaten will cause a change in the genetics of a population. You're going to have to blend in or run faster, something.
Adaptation.
Okay, these are all adaptations, so give me some specifics. Okay, feeding, okay, what you eat. So feeding strategies like the adaptation of bills on beaks on birds or their feet. Okay, so feeding strategies. What else? Yeah, so if you can live in the cold with big, thick skin or not, so that's good. Being able to live in the environment, like too cold, too wet, too dry, too windy. This one's a big one. We always forget it. Disease and resistance. Thanks for joining in with us. It's very nice. Disease resistance. Disease resistance. Guys, we have to stop there. Please study, and I'll see you on Thursday.